Hello and welcome to the second part of the lecture series on how the information gets out of our DNA and into the rest of the cell and tells the rest of the cell how to do everything it needs to. This step is called translation. We did transcription and now we get translation. In order to understand what happens during translation, you need to first understand something called tRNA. Now, we know that at the end of transcription, you had a molecule of mRNA that floated out of the nucleus through one of the nuclear pores and into the cytoplasm. mRNA is a long sequence of nucleotides, mRNA nucleotides, that are bound together. So you could have a sequence of nucleotide bases on here like A, U, G, etc., all the way down. tRNA is a little bit different. tRNA is shaped something like a T. It's also floating around in the cytoplasm. It has nucleotides all along it as well, but it only has 3 nucleotides that we'll ever talk about, which are here at the bottom of the tRNA molecule. Each tRNA molecule is also attached to a particular amino acid. And any particular sequence of 3 RNA nucleotides at the bottom of the tRNA corresponds to only one of the 20 possible amino acids. So an AUG, for example, might correspond to phenolphthalein. In order to simplify this drawing of the tRNA in the model that I'm about to draw for you, I'm going to simply draw a tRNA like this. Each of these lines at the bottom representing one of the 3 nucleotides at the bottom here. And then we'll put the letters at the end of each line. And then we'll label the amino acid that's attached to the mRNA, to the tRNA, up here, like this would be methionine. Now for the description of the steps in tRNA. Excuse me, not tRNA, in translation. First step, the ribosome binds to the mRNA. Now, if you remember, the ribosome is the cell structure that makes proteins. We're going to actually describe how the ribosome makes proteins here. So we have the mRNA here that came out of the nucleus and is floating in the cytoplasm. And what happens is that it binds to a ribosome. Ribosomes have 2 parts, a small subunit, which I've drawn there, and a larger subunit, which I've drawn here. The ribosome has particular areas in it, one here for binding one tRNA, and one here for binding a second tRNA. When the ribosome binds to the mRNA, it always binds to a spot on the mRNA that has the same sequence of nucleotides. At one end of the mRNA, you'll find a sequence AUG. That's what we call the start codon. It's the instructions from the mRNA that tells the ribosome to bind, and that's where it should start reading the message from the DNA. Now for step 2. This is the step in which the tRNA binds. So again, we're going to draw the same picture. We've got mRNA here, which we're simplifying slightly by making it nice and straight. We've got the small ribosomal subunit. We've got the large ribosomal subunit here. And then a tRNA molecule with a methionine amino acid, which we abbreviate MET, comes in and attaches to the mRNA. If you remember, the 3 nucleotides at the beginning of this mRNA strand are A, U, and G. The first amino acid that it brings in is methionine, which is brought in by a tRNA that has a U, an A, and a C as the 3 RNA nucleotides at the bottom of the tRNA. Now, these 3 nucleotides here that I'm circling on the mRNA are called a codon. A codon is like a word in mRNA language. On the tRNA, these three nucleotides are called the anticodon. The anticodon is three nucleotides that are complementary to the three nucleotides in the codon. Next, a second tRNA can bind to the ribosome, and if the sequence of nucleotides on the mRNA is UUC, then the next tRNA that comes in would have to be G in order to be complementary. So again, the codon here would be UUC, the anticodon is AAG, and the two sequences are complementary. This happens to bring in an amino acid called phenylalanine. Once these two tRNAs are bound, the next step is that the two amino acids bind together. If you remember, a protein is just a long string of amino acids. Now the methionine and the phenylalanine are bound together. The third step, I'll put on this diagram as well, is that the ribosome moves further down the mRNA and more tRNA molecules bind, bringing more amino acids in. The ribosome moves down the mRNA. It would move that way in this drawing. It moves in sets of 3 bases. In other words, one codon each time it moves, because the ribosome reads mRNA in 3-letter words, 3-letter chunks all at once. Now, for the fourth and last step, the ribosome has now moved down the mRNA molecule quite a ways. It's moving from right to left. This end of the mRNA is now free and floating in cytoplasm. There is a tRNA bound here with complementary nucleotides to the codon, and it has a long string of amino acids attached to it now that were specified by the bases in the mRNA molecule. The ribosome keeps moving down until it runs into this sequence, UGA. There are actually 3 sequences. That's one of the 3 that can tell the ribosome to stop. But UGA tells the ribosome to stop and let go. So step 4 is the ribosome stops and lets go. At that point, you have just this long string of amino acids. The order of the amino acids was specified by the sequence of bases on the mRNA, and this sequence of amino acids then folds up and makes a protein. The last thing that you need to know is how to determine what these amino acids are just based on the mRNA sequence. To do that, you use something called a codon table. I'm going to pull up the image of a codon table here. This is a codon table. What you do is look at the mRNA sequence and find the first nucleotide in the sequence. Let's say it's A. Then you find the second nucleotide in the sequence along the top here. Let's say it's U. And then you find the third nucleotide in the sequence on the right side. Let's say it's G. Right here. So you find the box in which all 3 cross. AUG is your first sequence of nucleotides on the mRNA, And that means that the amino acid methionine is your first amino acid. Using that method, you can determine the amino acids in order for any sequence of mRNA that you are given. That's it for now. Enjoy the problems.